This is episode 79 of the Immunology Podcast, ILC2s, the Microbiome, and Fibroblasts, with Dr. Kellen Cavagnero. Hey everyone, this is Dr. Jason Goldsmith and Dr. Brenda Raut. Welcome back to the Immunology Podcast. We have conversations with immunologists. The Immunology Podcast is brought to you by STEMCELL Technologies, a global biotechnology company supporting the life science research and fostering communication and collaboration in science. If you enjoy the Immunology Podcast, rate us and leave us a review. We're always looking for feedback on how the podcast can be improved and for suggestions on guests. Today we have Dr. Kellen Cavagnero from the University of California, San Diego, on the podcast to talk about his research on historically underappreciated players in innate immunity. We also got our usual roundup of recent highlights in immunology news coming up, but first...
If you're enjoying the interviews on this podcast, we invite you to read more interviews on the STEMCELL Technologies website. In the Immunology Profile series, immunologists tell their stories, discuss their research, and voice their thoughts and opinions on current topics in immunology. You can find all of these interviews at stemcell.com/immunoprofiles.
Well, so as people will hear in a minute, Dr. Cavagnero, Kellen, is also a podcaster. And it was fun to talk with someone who kind of knows the business and how to talk on radio, so to speak, or virtual radio, or whatever you want to call it. I think we both listen to podcasts. What are you up to these days in terms of listening?
Oof, that's a tough one. I've always liked podcasts, and probably one of the reasons that draw me to be doing this podcast here with you. I like to indulge on my interest in history in my podcast. So there's a couple of I like to follow this history podcast. There's one in particular that I really like, which is called "You're Dead to Me." It's from the BBC. It's from this guy called Greg Jenner. He's great, and he basically invites. This is a very common format, right? You have a historian and a comedian and the host, and they kind of talk about some particular topic in history, and then the comedian is always adding some little fun thing, because some of them know history, some not really. So it's a very fun conversation, and then at the end they quiz the guy, the comedian. So I really like that. I find that so entertaining, and that's probably one of the things and then I listen to a lot of very, I think more popular podcasts. This American Life and Radio Lab. These are things that are very well known in the US. I really like them. Oftentimes they have topics that are super interesting. And then I also listen to podcasts to help with my languages. So I have a couple of German podcasts and Dutch podcasts just to try to keep me practicing that. No podcast in Spanish though. I should find one so I don't forget. I should get an Argentinian one. Otherwise I will lose my accent. That will be horrible. My accent is very important. It's part of my identity, Jason. Shame on you. What do you listen to?
Mostly economic podcasts. So Make Me Smart from Marketplace, Marketplace Tech, Planet Money.
Oh, I like Planet Money. I also used to listen to that. It's not so economic. They also have more everyday stuff.
The Indicator, Freakonomics.
Is that still going, Freakonomics? I also used to listen.
It is, and I've listened to some other stuff like No Stupid Questions from Freakonomics. There's Freakonomics MD for a while. I listened to a few of those. That was fun. And then I occasionally listen to Joe Rogan. It really depends on the guests that he has. Like when Neil deGrasse Tyson's on, that's fun, right? So he'll have some interesting people, or since, I do MMA as well, he'll have some interesting famous fighters on that I think will be interesting to listen to. And then I don't listen to it for other times. Those are the big ones. There's obviously some NPR, like NPR Politics and 1A occasionally. Depends on how much lawn mowing and yard work I have in a week.
Yeah. Now that some time ago, I moved closer to work. I used to bike half an hour to work. So I had a lot of time for podcasts. Now it's a little bit less, less than a 10-minute bike. That removed a lot of my podcast listening time.
Yeah, I had a train ride I used to, and that added a lot of podcasting on, but no more train for me.
Well, we'll see what our conversation with Dr. Kellen Cavagnero from UC San Diego will bring us. But before we get to that.
So the first one up I have is in immunity. It's titled breadth of FC-mediated infection function correlates with clinical immunity following human malarial challenge. 2 first authors, Irene N. Nikumama and Rodney Ogwong. It was published on May 23rd. And so this paper makes a point that I think helps with drug and vaccine design for malaria. And they, unknowns to me, apparently what they use in the field a lot is this growth inhibition assay to evaluate, prioritize, and determine, hey, is this a good vaccine or not? And a problem with malaria vaccines as well is that they only typically work on one part of the life cycle of the vaccine. And so since you have different antigens at different parts of the life cycle, it's easier. That's why it's hard to vaccinate against malaria, is it has all these evasion mechanisms just by being in its different form, kind of like Pokemon. So there you go. So it gets harder to do. And they have, what they did is they have controlled malaria challenge studies in semi-immune volunteers is something that's been done to try to understand what really correlates with mechanisms of protection. And what they do in this paper is show that functional antibody assays, which I'll go over in a second, because this is largely a methods paper and a way of proving something, are better than growth inhibition assays in evaluating if something is a good vaccine or not, or if you're immune or not. So what do I mean by functional assays? Example of things like, you have this growth inhibition, but that's not a good one. Antibody-dependent complement fixation, opsonic phagocytosis, activity of merocytes, phagocytosis of ring stage parasites, antibody-dependent respiratory burst, things like that, right? So they're looking at if you have these antibody-mediated immune responses, FC, can you measure that in vitro with the patient's blood or what have you, and malaria, and assess, is that better? And the answer is yes, fundamentally. And so they take these people that are somewhat immune and non-immune cohorts, they get infected, and the people who are immune, they'll have similar results on this GIA measure, but the people who are immune versus the non-immune arm had much better FC mediator function, and it had a much better prediction of what the patient actually was, either partially immune or not, and how long, if they got sick with this challenge, were they sick. Not only that, but even in people who were partially immune, how long they were sick was predicted based on their outcomes of these assays. And they also show it's not just about antibody binding, but specifically this FC effector function, right? So it's not just antibody strength of binding, but these effector functions. And so the point of this entire paper is really, instead of doing this quick growth inhibition assay and say, oh, we have growth inhibition with this vaccine, we should use it, you're not actually measuring something that has clinically meaningful output. And that's what this controlled infection study showed, is those people who are immune and who resist repeat infection in that cohort, they have these effector function changes. And those are the assays you should use to decide if your vaccine is doing something useful clinically. And so I think it really goes to the point that there's a really important part about translational medicine and science. Hey, plug for us people who are overeducated and decide to do both, that you have to measure the right thing to assess if it's really going to work or not. And having the right measure up front is going to help a lot with downstream success. So there you go. That's the first paper I got.
Okay, very interesting. I guess there's also a little you can learn from this challenge assays that you cannot really learn from just following the infections in normal people. It's very interesting.
Yeah, you're kind of pre-selecting for the outcome you want and then back calculating what's a good measure of that outcome, which I think is powerful.
All right. So for my first paper of the day, I want to share also a story, about fighting infections, translational medicine. So this is, isn't all medicine translation in immunology. This paper is called Efficacy and Safety of Novel Multifunctional M10 CAR T cells in HIV-1 infected patients, a phase 1 multicenter single-arm open-label study. This was published in the journal Cell Discovery, which is a journal that is sponsored by the Chinese Academy of Sciences together with Nature, and therefore it's not surprising that the work was done at the Fudan University in Shanghai in China. First authors, we have a many first and many corresponding authors. So first authors Yunju Mao, Qibin Liao, Youwei Zhu, Mingxuan Bi, and Jun So. And then the corresponding authors are Renfang Zhang, Jing Leng, Zhang Taozhen, Xiaoshan Zhang, and Xianqing Zhu. I really hope that my pronunciation made sense. Basically, the idea of this study is looking into the trying to use CAR T cells for fighting HIV infections. So the idea, so HIV, of course, is still a significant health challenge in the world. But well, we do have antiretroviral therapy, but that can kind of keep levels of the virus very low and provide a more or less normal quality of life to patients. But this therapy does not eradicate the viral reservoir. So that's why people need to be on antiretroviral therapy for the rest of their lives if they're infected with HIV. So in this study, they're exploring using chimeric antigen receptor T cells or CAR T cells, and they make like a new approach at how to design these CAR T cells, trying to target HIV-1, and particularly like target the reservoir of the virus. So they're not the first people to think about this. So there have been previous studies looking into using CAR T cells that are detecting some protein from the envelope of the virus, and then they could potentially kill infected cells. But what I think is interesting is that they try to kind of add more skills to these cells, not only their CAR. So they develop these cells, which they call M10 CAR T cells, that are basically are engineered to have three functions. On one hand, they have kind of broad cytotoxic effects on HIV infected cells because of the chimeric receptor. They are expressing a receptor that has two different specificities that can that I will give more a little bit more detail in a moment. They the cells also have a construct that produces free broadly so free antibodies. So they also secrete broadly neutralizing antibodies, and they are designed to home into a B cell follicle, which makes them easier for them to go and target the viral reservoirs in lymphoid tissues. And so they do this by adding the expression of CXCR5, which is this follicle homing chemokine receptor. So when it comes to the specificity of the CAR, I think it's very interesting because they have this CAR receptor that is targeting two different epitopes on the envelope protein of HIV. On the one hand, they have this site that binds to this is a receptor that binds. It's a mutated version of the CD4 receptor, which is targeted by the envelope protein when it's binding to enter the cells. And on the other hand, they have these exposed, like this modified CD4 epitope that only occurs when there is an initial binding of the virus, and then there's a conformational chain that exposes a site that is also bound by the envelope protein. And though this receptor that they have has both of these things, so in principle it can bind to the envelope protein through two different sites. And when they come to these broadly neutralizing antibodies, these are targeting this also another part of the envelope protein of HIV, and is a particular site that is capable of neutralizing cell-free viruses and block their ability to infect other host cells. So these two things together kind of, on the one hand, targets infected cells, on the other hand, prevents viruses from running around infecting new cells. So I thought it was very interesting. And when they also add this CXCR5 receptor, this is with the idea of getting the T cells to the right place to target and to select, to find infected cells that are part of the reservoir of the virus. So what they do, so they design this. They have this cells. They do a lot of in vitro analysis of these cells. They show that indeed they produce the neutralizing antibodies. They are binding. They can bind to the receptor. They show that they can migrate through their signaling through CXCR5. So they have all these preclinical assays which are satisfactory. But I think what is very interesting, I don't think that is done in many places, is they actually do a phase 1 trial. So they have 18 infected patients who are on ART, so antiretroviral therapies, and they treat them with an infusion of these CAR T cells made from PBMCs from HLA haploidentical donors, which means that it's either a son, like children, or parents that have exactly half of the HLA alleles is the same. And this allows to improve kind of survival and engraftment of these cells. And so these people, they infuse 2 infusions of these empty CAR T cells with 30 days in between. And then what is interesting, and this is something that has already been discussed in previous research, is that they give a compound called ketamide, which is a latency reversing agent that reactivates the virus. And so if you administer this to patients, you see a peak in viral RNA in the patients, and in this way, they're looking into exposing infected cells to these CAR T cells. And I think that overall the results were fairly promising. So they do see that having these M10 cell infusions led to significant suppression of the rebound of the virus in like almost 3/4 of the cases. And they see viral load reduction. And this is particularly kind of evident when they do the second round, like when they do a further reactivation, that they see that with a second reactivation, the amount of virus that comes out is lower than the first time, suggesting that the reservoir, the size of the reservoir is diminished compared to the initial situation. And they see this, they see that in some of the patients that have been treated for already, almost 6 months, no, so for 150 days, they see that this reduction is sustained. So I thought, and also they don't see significant adverse effects related to the therapy. So I think it's very interesting because, the idea is that by doing this and forcing the reactivation of the virus, then you can expose cells that are reservoirs, and because you have these neutralizing antibodies, you're kind of preventing further dissemination of the virus because you're neutralizing it as it's coming out. So I thought it was a very interesting approach, and at least this initial small study seems to be fairly encouraging.
Yeah, I wonder if it'll get someone to be all the way seronegative, if it will result. Is it fundamentally better than just being an antiretroviral therapy for your life? Because CAR T cells do have, maybe some risks, right? Maybe some cancer risk and stuff. And if you have it in your body for 40 years because you have HIV or whatever, that's a bigger risk than taking it in your 60s or 70s to prevent your terminal cancer, especially if heart's available.
Yeah.
So that's where my brain goes on this one.
I completely agree. And I think this kind of makes me think of that, the Chinese, that rogue Chinese scientist that made the 6ER5 mutant babies, the CRISPR babies. So it really makes you think, what artworks people, do become undetectable, and it's not that hard and it's not that difficult to stay on this therapy. So yeah, it does feel like it's a little bit of an overkill. Um, and we're not completely sure what are the risks long term of of CAR T cells. I mean, if the in a case of leukemia or severe lupus, the alternative is you die. But in this case, yeah,
Yeah. Now, if there wasn't heart, then a different story. But yeah, I wonder about that. And heart
yeah.
has side effects, but I just wonder the risk versus reward, and thus the regulatory pathway for this could be tough.
Well, I have to say, I don't think you could do this clinical trial outside of China. I don't think you could do it in Europe because exactly people would say this is too risky for the benefit.
Yeah.
I wonder.
All right. Well, shifting gears entirely, I'm going to go way into the weeds on cell death here and try to just give the high level view. So also in immunity, titled a RIPK1 specific PROTAC degrader achieves potent anti-tumor activity by enhancing immunogenic cell death. First authors are Jonathan Mannion, Valentina Gifford, Benjamin Bellini, and Winnie Fernando. And corresponding authors are Tencho Tenev and Pascal Meyer. All right. Also published May 23rd. So RIPK1 is a scaffolding protein that's a kinase and has these dual functions. It protects cancer cells from immune detection and death by blocking RIPK3 and then necroptosis from MLKL. It also, though, blocks NF-kappa B activation, and NF-kappa B can be good or bad, right? Like, so it could be pro-survival, it could be pro-inflammatory, pick something. So PROTACs are a system of you tag a protein, it binds to a protein, and then has a ubiquitin ligase homing ligand on it or motif on it as well. And so it targets proteins for targeted ubiquitination and degradation, right? So it uses your endogenous system to degrade a protein. So they made a RIPK1 PROTAC to see what you could do therapeutically to do this, because just knocking out RIPK1 leads to all types of bad things with unfettered necroptosis. That ain't good. And so they want to see what happens. So they sure show that, hey, their PROTAC works. Congratulations. Okay, yay, their PROTAC works. And if you use it, it makes innate immune signaling wacky. It compromises TNF and TLR3 signaling. And it works differently in different cells somewhat. So it dysregulated responses in primary BMDMs and triple negative breast cancer cells, and enhanced TNF cytokine expression, even though you wouldn't necessarily expect that. So it's accentuating NF-kappa B dependent, TNF NF-kappa B axis signaling, while simultaneously leading down a necroptosis pathway. So you kind of have this inflammatory NF-kappa B thing and a necroptosis pathway. And they're like, it kind of has competing functions because it's dual inhibitory of things that are competing. And so you remove it and you get dual activation of competing things. But the question is it useful? So they first do some signaling work and show that, look, it works through TLR3 and 4 and TNFR1 mediated activation of RIPK3. So the RIPK1 protects by getting rid of RIPK1. That's how it functions. And then they deep dive in, and it's independent of TRIF and ZBP1 for people who know all the signaling. And they do a bunch of work to show that, because all these pathways get very intertwined. And by doing this, it helps achieve anti-tumor immunity. And so then when they go into cancer lines, you get more necroptosis by the immune system, driven by the immune system. It sensitizes cancer cells to TNF and interferon related signaling by getting rid of this, you can have better killing. And then they do that through a radiation system. So they do orthotopic injection into mammary fat pads of triple negative cancer. And they show that, hey, with radiation, you're getting a better response. And then they also go and do, I believe, immune checkpoint blockade and show that you're getting better response there. And then they look at a skin inflammation model that's known to occur spontaneous RIPK1 knockouts and show, or not in RIPK1 knockouts, RIPK1 activation mediates this through a caspase-8 deletion. And so if you get rid of RIPK1, it inverts that, and then they do that with this drug. And so they show that other signaling known to be RIPK1 dependent, which includes autoimmunity, if you get rid of RIPK1 through this drug, that also has an effect there. And so it's more of a proof of concept of a protein-based therapy that could work, right? These are protein-based therapies, but also, but going down this pathway to hone cells to be more sensitive to necroptotic signals and inflammatory signals by NF-kappa B activation through removing this dual inhibitor, but then simultaneously in other systems with balance where you can have autoimmunity due to caspase-8 deletion, or RIPK1 is then driving the autoimmunity, getting rid of that turns off the autoimmunity. So cool mechanism for degrading a protein, because SC versus siRNA knockdown, it has pretty nice kinetics. And they got somewhere with it. And that was fun.
Can you remind me what is the proteolysis targeting part of this molecule?
So it's a dual tagged thing, right? So it has a binding domain that a linker, so a PK68 based RIPK1 binder. So that's going to bind, that's like a ligand target that's going to pull in the RIPK1 as a linker, and then a VHL von Hippel-Lindau E3 ligase binder.
Oh, okay.
And so you have this strong linker the strong binding ligand with a linker to this von Hippel-Lindau E3 ligase, and so then that causes ubiquitination and proteasome degradation of RIPK1 through the endogenous proteasome.
And so, and these molecules all they just how do they enter the cells?
Oh God, how did they do this? I think it gets just, or they don't put it in a liposome or anything. I think
Exactly. That was my question.
They just inject it.
Hmm.
They just put it in the tissue culture, from what I understand.
Okay. It just gets in somehow.
I think it's small enough that it gets in on its own.
Okay.
Oh, they injected solubilized encapsitol and administered by intratumoral injection. It seems to bypass the cell membrane.
Yeah, but it has to get into the cells somehow.
Right. So it gets through the cell membrane on its own as a drug.
Okay.
I think maybe that von Hippel-Lindau thing gets pulled in, but they're not big. They're not huge macromolecules, right? It's a small double-ended linker, but somehow it gets in.
Okay. All right. So for my second contribution to this conversation, I have a paper about autoimmunity and where it comes from. Terrible segue. And this paper is called NUDCD3 Deficiency Disrupts BDJ Recombination to Cause SCID and Omen Syndrome. And this was published in Science Immunology on the 24th of May. First authors Rui Chen and Elena Lukianova, from the corresponding author Sophie Hambleton at Newcastle University, and also I think it's a big consortium. And I like these papers because this is the typical example of, you start with a problem that is like very patient centered, and you try to figure out where it's coming from. You use some nice genetic models, and then you reach kind of a conclusion. So in this study, they're looking at patients that suffer from this autoimmune and kind of immune dysregulation syndromes called SCID or SICD, I guess, and Omen syndrome. And so SICD stands for severe combined immunodeficiency. And basically, It encompasses a group of fairly rare but life-threatening diseases that are characterized by severe defects in both T cell and B cell lymphocyte development. And this generates kind of, on the one hand, um, it's paradoxically you have autoimmunity, but also susceptibility to infection, and basically it can be very lethal. Patients can be treated with bone marrow transplantation, for example, and therefore nowadays more people get genetic testing for known sources of this SCID. For example, deficiencies in an IL-2 receptor or RAG1 or RAG2, which are important for recombination of the TCR and the BCR during development. And Omen syndrome is a subset of this SICD, which is particularly characterized by some defects in their combination of of immune receptors of B cells and T cells, usually due to the a deficiency in RAG1 or RAG2, but also other DNA repair genes that are involved with this process of mutation for recombining these chains of the T cell receptors and B cell receptors. And again, these patients have a lot of autoimmunity, inflammation, but also immunodeficiency, and it's quite severe. But so there's a lot of identified genes that are affected during this disorders. But what they did in this paper is they looked into patients without a known cause for disease, or without a known without knowing exactly which is the gene that is at fault, trying to find where the deficiency was coming from. So basically what they do is they do a whole exome sequencing, and they compare. They have clearly some kind of consortia where they have data from a lot of patients, and they have whole exome sequencing from all of them, and they find in a subset of these patients a single homozygous missense variant in this gene NUDCD3. I want to say NUDCD3. That stands for NUDC domain containing 3. And so it does segregate with the disease in a series of family trees of people affected with this disease. But this in general is a very rare allele. So not really observed. And this in particular is in a subset of patients with Asian descent or Asian ethnicity. And when they look at this gene, this mutation, they see that it generates some kind of loss of function, and there's this G that is exchanged by an A, and this so basically an aspartate is replacing a glycine at this position 52, and this is in a highly conserved N-terminal domain of this protein. So basically, this does seem to be like the mutation behind at least this subset, that has all these patients have in common. And not much is known about this protein, especially in the context of immune cells. But they do know that it does exhibit some kind of chaperone activity, and it has been shown in other systems that it chaperones proteins. But there was one reported appearance of RAG1 as in the interactome of a NUDCD3. So that, I think that's what really got them interested, seeing that there was some kind of association with RAG1, which is a known gene involved in the development of these SCID diseases. And they find that NUDCD3 is expressed in the thymus and in the bone marrow. So they do think they're up to something. Basically, so they study cells from patients that have this variant, and they also have a mouse model in which they introduce this mutation in these mice. And basically, they do show that indeed there is a inhibition or there is a deficiency in the recombination of both T cell and B cell receptors. That is in a way comparable to people that have Omen syndrome. So again, I'm pointing to deficiency in the functioning of RAC1 or RAC2. And I think probably the most interesting, so they do, I don't want to go too much in details because they do a lot of assays in which they're trying to characterize where is the problem happening. So they see, um, uh, they kind of show that that indeed they have a lot of ways of justifying that there's an issue at the point of recombination. And there's many clues about this, for example, in which part they see a particular subset of like developing T cells, it's thymocytes actually that get accumulated in thymus that are the ones that are right before, proper recombination, suggesting that there is a bottleneck at the recombination step. They show that the clones that do exit the thymus have uh, TCRs that are very different in their characteristics to normal people, normal TCR output, particularly when it comes to the clonal type. They have a lot less clones, a lot of expanded clones, and the usage of the genes, the different gene parts, the segments that are recombined to generate a functional T cell receptor have the telltales of a defective or subperforming level of recombination. So there's a lot of things that really show that this is where the problem is. And so the question is, why is NAT CD3 affecting recombination, and what does it have to do with RUG1? And I think that's a little bit more interesting. They don't see differences in localization of either RUG1 or RUG2 when it comes to the nucleus or the cytoplasm, but they do see something that I wasn't really aware of, is that within the nucleus, there's also this nucleoli localization of RUG1 particularly that happens throughout development, and that it's important in order to properly time the action of RUG1. And so what they conclude is that they see that the localization RUG1 seems to localize more within these nucleoli when you have this mutant variant of NAT CD3, and that there is some kind of dimerization of NAT CD3 that is not properly happening, that is required in order to kind of chaperone RUG1 out of these nucleoli structures. And so by preventing the nuclear egress of RUG1, which is dependent on NAT CD3 function, then you don't have RUG1. Kind of RUG1 is in a way mislocalized and cannot properly carry out the recombination required for these T cells. So this is basically the conclusion that they say, and how they show using a lot of cell models. They also do some heck experiments, and this is the conclusion that they arrive. And now it's very interesting because starting from just a whole exome sequencing of affected patients.
Oh, I like you got the mechanism there at the end. You were making me hold out.
Yeah, I know. I leaving the best for the last.
Well, we don't want to hold out the audience any longer. Ho, segue. And we're going to be talking to Dr. Kellen Cavagnero at University of California, San Diego in just a moment. But before we get to that, are you performing multiple rounds of cell isolation? That can be inefficient. And I love efficiency, and I love to see Brenda getting out of the lab before midnight. Using the new EZ250 EZ-SEP magnet, you can scale up your cell isolation and process large volume samples like leukoplex and whole blood in a single round of preparation. Obtain highly purified cells from samples of up to 225 mils in a single step. Learn more at stemcell.com/EZ250magnet.
For today's interview, we are joined by Dr. Kellen Cavagnero. He is a postdoctoral fellow at UC San Diego in the US, and he is one of our first trainee guest that are going to be in our podcast to talk about his research. He recently defended his PhD thesis and also about his experience podcasting, as such, like us, and the new experience of transitioning from PhD to postdoc. So, dear Dr. Cavagnero, thank you so much for joining us. Welcome to the Immunology Podcast.
Thank you very much, Brenda and Jason, for having me. It's really an honor to be included in this podcast with all of your esteemed guests. So thank you so much for having me.
You're most welcome. We're looking forward to it. So let's start with like the rear view mirror a little bit. You just defended. So what was your work on, and what was it like to finally be done?
Well, I'll start with the last part. It feels really good to be done, for sure. Feels really good. As far as my work, I guess I can start with really the background and how I got into it. So many people listening are probably familiar with Staphylococcus aureus or Staph aureus, a bacterial pathogen. It's the major cause of skin and soft tissue infections in humans. It's most people who get a staph infection, I've had a couple myself. You take antibiotics, clears right up, no problem. But we have really a big problem with antimicrobial resistance right now, with all of these pathogens, including Staph aureus, developing resistance to many of our antimicrobials. And so, this pathogen causes a lot of morbidity and mortality. Like about 20,000 people annually will die from MRSA infections. And so it's really important that we understand how our skin defends us against Staph aureus. And so really that's what my PhD thesis set out to address. So in terms of what we know already about how the skin defends us against Staph aureus, most immunologists really think about three major cell types. You have your keratinocytes at the skin cell surface. You've got neutrophils that will come into the skin to really very potently kill the Staph aureus, those professional killers. And then the IL-17 producing lymphocytes in the skin that are going to really potentiate the keratinocyte and neutrophil mediated host defense. But we all know that there's many other cell types in the skin, and we really ignore a lot of them. And so what I really sought to address was use new technology that we now have available to take an unbiased look and see what are all these other cells doing in the skin for defense. And so I first started using, as most people do these days, single cell RNA sequencing. And it's not something I actually did a lot of before graduate school. I was more of just a bench immunologist, but I've become very immersed in the bioinformatic technology. And I think that's a really important thing for, you said you have a lot of trainees listening that are in the PhD stage or earlier. I think it's important to invest time in those technologies. And what I was able to find using this unbiased approach was that really a cell type called the fibroblast was doing a lot of things that could be very important for defense against Staph aureus. And, these cells are beneath the epidermis. They're in the dermis. They make up the major cell population there. They're mainly appreciated for making extracellular matrix and providing structure to the tissue. And they've kind of been historically ignored in terms of immunity and everything else. And what we found doing the single cell RNA sequencing was that not all fibroblasts, but just subpopulations of fibroblasts made a lot of neutrophil chemokines and antimicrobials, suggesting that they may perform similar functions to those keratinocytes at the cell surface in terms of killing the Staph aureus and recruiting neutrophils into the skin. And so we validated this with other approaches like confocal microscopy, and we ended up what we call these cells now is immune acting fibroblasts. We think it's important to define these subset of cells as a unique entity because it's really tough to work on something if you don't really know what to call it. So we think it's important to call these things something else rather than just fibroblasts, and to distinguish them as immune acting and highlight their role or potential role. And so, it looks like these cells were doing important things, but we don't really know if they actually are. So we did a lot of in vitro work, in a dish in culture, showing that fibroblasts respond to not only pro-inflammatory cytokines like IL-17, but also recognize bacterial products like toll ligands. And so these cells are potentially sensing signals sent by immune cells and by microbes and eliciting important immune functions. And we showed that these cells are very potent recruiters of neutrophils using in vitro chemotaxis assays, and worked out all the mechanisms that they're doing this through. And then the icing on the cake really at the very end was we developed conditional knockout mice that lack specific receptors in the fibroblasts that limit their ability to respond to these pro-inflammatory signals like IL-17. And these mice had impaired host defense against Staph aureus because they had really an impaired ability to recruit neutrophils. And so we really identified a new player in host defense against Staph aureus, and that was these subsets of immune-acting fibroblasts. And because the effect of IL-17 was so strong on these cells, we wanted to look at another disease that's well known to play, where IL-17 is known to play an important role in, and that's psoriasis, which is another skin disease. It's not an infectious disease like Staph aureus infection. It's more of a inflammatory skin disease. And so we used our conditional knockout mice in that model, and we also saw very significant reductions of neutrophil recruitment to the skin. And so these cells are important not only for defense against Staph aureus, but also in non-infectious inflammatory contexts like psoriasis. And we also, the final thing that we did in the paper and in my thesis was show that this is relevant in humans using samples from patients that are on and off IL-17 inhibitors. And in the context of IL-17 blockade, we see that fibroblasts and their expression of these immune factors like neutrophil chemokines was severely suppressed.
I like how we are evolving our understanding of fibroblasts from just, cells that are just there to really players in several different contexts. Also, I'm now thinking about all those cells in tumors and how they are intervening in the immune response, things like that. I would argue that this is one of the, Jason often says, that enterocytes are immune cells. I guess that fibroblasts are now immune cells too. Jason is not convinced. What do you think?
No, I love it.
Everything.
If you got a TLR on you, and you single chemokines or cytokines, and especially if you have antigen presenting HLA, what else are you but an immune cell?
Yeah, I don't think these fibroblasts are either the MHC1. They're not really presenting other type of HLA. I assume.
But if you got TLR signaling and chemokines, you're getting pretty close.
Yeah, everything is an immune cell. It's just.
All right, Kellen, are fibroblasts immune cells?
I think so. And one of the members of my committee is Chris Glass, who's a macrophage expert, and I think he really was interested in my work because these cells seem very similar to macrophages, just fibroblasts as a whole. You have these cells that can be very plastic and polarized into these two categories where you have the sort of defense properties and also tissue repair properties. So I think if you think of a macrophage as an immune cell, I think a fibroblast should be an immune cell too. It's not hematopoietic, but I think it's serving many of the same functions.
That's my view. Enterocytes are immune cells for the same reasons macrophages are. They're just not hematopoietic.
Yeah. I mean, there's other immune cells that are not every immune cell is hematopoietic, right? All these fetal derived cells they're not hematopoietic, are they?
No, kind of. The stem cell compartment moves. So that's a really philosophical question there.
They express CD45, right? So that's
yeah. Oh, all right. Yeah, I guess.
Yeah.
That's an easy line to draw.
There we
go. All right. So I don't think you mentioned. So this is these fibroblasts are making CXCL12, right? And they are the one, and that is the chemokine that is recruiting these neutrophils.
So they express several chemokines. They express the CXCR2 ligands, which are 1, 2, 3, 5 in mice, and then you have the others up to 8 in human. And they also express CXCL12, which is a CXCR4 ligand. And in vitro, we could completely explain the fibroblast recruitment of neutrophils through these CXCR2 and CXCR4 ligands. When we inhibit them, everything goes away. In vivo, we haven't fully elucidated the role of each individual chemokine. We opted to demonstrate the significance of the fibroblast in vivo, knock out the IL-17 receptor, which was really a key activator of the fibroblast. We have not done the conditional knockouts yet for the specific chemokines. This is something we're working on. I anticipate that it's going to be a little bit complex because of redundancy, possibly. In terms of the fibroblast subset that we identified, though, CXCL12 was the broadest marker of the fibroblasts that were doing the immune cell recruitment in the single cell, whereas other cells, like there were subpopulations of CXCL12 positive cells that expressed other chemokines like CXCL1. So essentially, it's complex, and there's what I'm trying to divide it into right now is CXCL12 positive, which is the adipocyte lineage neutrophil recruiting population of fibroblasts, and then the CXCL12 negative, which is more of the structural ECM scar-like fibroblast. But then within that CXCL12 positive population of fibroblasts, there's many more subpopulations that express different levels of other chemokines. We haven't really defined those at a more granular level yet, but that's something I think is important to do going forward.
So you finished your grad school, but you're staying at the same institution for now for postdoc. Are you switching labs? What's your plan? Are you going to keep going on this with some follow-up studies? Make a run for it?
That's the $1 million question. That's the $1 million question. So I've actually been at UCSD for almost 10 years now. I did a very long gap year of 5 years in between my undergrad and graduate school. I went to undergrad at UC Santa Barbara and finished that in 2014. And then I moved back down here where I grew up in San Diego. And I joined a lab, working as a quote unquote technician in the lab of Dr. Taylor Doherty doing lung immunology, studying group 2 innate lymphoid cells, just doing lots and lots of flow cytometry back then. And I really enjoyed the lab, and I ended up sticking around in his lab for about 5 years. And it felt in very much in many ways my first grad school experience. So I was writing papers and leading my own projects, and then after 5 years, I decided, I think I kind of need to go to grad school because I think I really like this academia thing. And so then I was fortunate enough to get into UC San Diego and joined Richard Gallo's lab, which is where I've been doing all the work that I described previously. And I also need to acknowledge him and my co-authors for all their help with that project. It was really a group effort. So now in terms of postdoc, I feel like, I've gotten a lot of different opinions from different people about what I should do. And I've thought long and hard about what I should do. And I'm leaning towards staying in my current lab at UCSD. And this is controversial, I guess. Sometimes when I tell people this, they kind of make a face at me like, you're gonna do that. But I guess the rationale for changing labs is to get a diversity of experience and perspective. And I feel like I do have a bit of that, given that I've been in 2 different labs already and have, 10 years of experience. And I'm kind of eager to go to the next step. And I'm fortunate with my current mentor, Rich Gallo, who's, I think he's graduated something like 30 plus grad students and postdocs, and 20 of them are in academic faculty positions. So he's really been a great mentor for many. And he's been very supportive of me and believing in me and helped me map out a path to become an independent investigator. And I think I'm going to continue to work with him to make that happen at the moment. That's where I'm currently at.
I mean, if you have a project that you could take with you, which it sounds like this guy or your PI knows how to do that, right? That's a big thing. Because sometimes you have to take like your grad school, go to a postdoc lab, and then because of the mix, you can take it with you. But if you have something you can take and that's identified, then there's less of an impetus to move. But if you wanted to stay at UCSD as faculty, that would be harder by doing a postdoc there.
Yeah, I'm taking it one step at a time. I don't know exactly. I love the West Coast. I grew up in San Diego. I love to surf, and I love just the weather here is amazing. It'd be hard to move somewhere else just for the sake of moving. But when it comes to getting a faculty position, I think I'll cross that bridge when I get there. It's a long ways away. But in terms of the project, so the fibroblast thing was really pretty new for our lab. We had an interest in stromal cells before I got to the lab. We had discovered this phenomenon called reactive adipogenesis back in 2015. I can describe that briefly. So in Staph aureus infection, a postdoc found that when you give a mouse like intradermal infection of Staph aureus, that subsets of fibroblasts that are pre-adipocytes will turn into fat cells And while they're turning into fat cells, they make antimicrobials, and this is essential for killing Staph aureus. And so a lot of people in the lab are really focused on this reactive adipogenesis process. I know, Jason, you're a gut guy, right? We have a gastroenterologist in the lab who showed that reactive adipogenesis also occurs in the gut in the context of inflammatory bowel disease.
Oh, yeah. You get all this fat stranding, and you see it like if you go and do surgery on them afterwards.
Yeah, and we showed it happens in the very common skin inflammatory disease, acne. And so a lot of people are focused on this, fat cell differentiation process. And my work was really different in that it was independent of fat cell differentiation. It's just the fibroblasts in their undifferentiated state are doing this host defense process through neutrophil recruitment. And so, and Rich has been very supportive of me taking this work with me. But in terms of the postdoctoral training, I think what I would like to do going forward is had another mentor come on board that can help me take this work to the next level, whether that's through new technology or a new disease context. Like I mentioned, I've done a lot of work in the lung before, and I've thought about, exploring fibroblasts and their host defense properties in the lung, or, completing the trifecta and getting experience in the gut, do all the barrier tissues. So there's a lot of I think room to grow these projects, and I'm still in the very early phases. As I mentioned, just ended last month, so still thinking about everything and then what I want to do next.
I find the discussion about whether to stay or whether to go, or this idea that you have to have, you need to leave for your postdoc. You need to. I've had, I've been working in, I've done research in 4 different countries. I moved for my postdoc and moved for my PhD. Um, there's also a penalty for leaving, because every time you have to start over, that also takes time, and also so sometimes I think it does make sense to stay, even career wise, because you can accomplish a lot more if you're already settled and you already know how things work and people know you, and you're familiar with what you're doing. And so I think it looks to me like you know what you're doing. So
I have
um,
no idea what I'm doing.
yeah, well, you just have to, how was the word, fake it till you make it. I guess that's half of the work.
No one
uh
knows what they're doing. We just keep going, and it looks
mm,
like we do. That's the secret.
secret, dear listeners. But I wanted to shift gears just a second here, because I wanted to talk about some of your side projects, and I think that's something that we have in common. So you have been hosting also a podcast called Inflammatory Content, in which you also are talking about immunology research. You have had several guests. You have a more of a long format, so a little bit longer conversations with immunologists, and I thought it was very interesting, especially for those who want to be inspired about ways of doing some kind of outreach communication. So why don't you tell us a little bit about what encouraged you to do this podcast, and what was the idea behind it?
Yeah, I'm happy to share my experiences with the podcast. So I love to teach, and when I was in that previous lab before my grad school in Taylor Doherty's lab, I was fortunate to have the experience to teach many undergrad and master's students, and I really got a lot out of that. And when I became a grad student, I wanted to try to do something else, and I listened to a lot of podcasts myself at the time. Some really great ones. There's even some really great science ones. I don't know if you listen to them, like This Week in Virology. Some really fantastic podcasts out there that inspired me a bit. Like, what if I could take my teaching that I'm doing one-on-one and really make it more accessible and really everlasting, because you put something out there on the internet and it can reach many, many people, and it can be up there forever, which is also kind of the scary part. And so I was giving a lot of journal clubs for my lab and for classes of the first year grad school classes. And I just decided, let's just record them on my phone and upload them on the internet, see what happens. And turned out that people were listening to it from all across the world, and were emailing me, asking me to questions about the science and asking me to do certain topics that they wanted to hear about. And so that feedback really inspired me to keep going. And I really like the podcasts that do interviews. I think they're also more fun to listen to and to actually produce as well. And so I started with a really talented postdoc in my lab, Alan O'Neill, and we had a great conversation, put it up there, and I was really jazzed about the idea of doing more. And I asked my PI if he'd like to come on, and of course he had to, because I'm a student, right? And it was a really fun time. And then I asked my committee members and then other faculty members that I know around the Torrey Pines Mesa, like, you know, we have UCSD, La Jolla Institute, Sanford Burnham Prebys, Salk Institute, all these great science institutes in this area, and many immunologists. And so just had some really great conversations, and kind of just started snowballing a little bit. I mean, my podcast is you know, tiny compared to your guys, which has such huge reach, and you know, that's why I'm so honored to be on here. But it's been a really fun experience, and I don't think of it as work. I find joy in doing it. The editing process can be a little bit cumbersome, but I really am grateful for what it's provided me in terms of, I think every conversation I have with people, I create this new bond with that person, and I feel like I'm getting a new mentor every time I talk to someone, because we have this shared experience. And so it's not only great for me in my career, but I think it's really great for the listeners too. And like I said, it's free, it's very accessible, anyone can listen to it anywhere. I'm sure with AI, we'll eventually be able to translate these things into any language, and they'll be able to get everywhere, and get into everyone's ears. And so I think it's a really great platform for communicating science, and I think it's really only the beginning. I think there's a lot more we're going to see from podcasting.
So you got your podcast, that's going. You're figuring out life with fibroblasts. As someone who just did a dissertation, and you know, you had to put that entire document together, and you probably contemplated all the things you did a year or two ago that you wish you had told your younger self not to do that way. Any like top 3 or top 5 list to grad students from someone just finishing of like, you know, I really would have put better stuff in my notebook or whatever, that like lessons that like, no one's going to listen to the PI on this, but you just were there, you just lived it a week ago. Any top 3, top 5 list of things you think you should pass on?
In terms of the dissertation and defense, I'd say start early, you know, start putting things together, work on your presentation. I think the defense was my most, I love giving talks. I love talking. And I still was so nervous for my defense, so nervous. And fortunately, I'm the kind of person who panics early. And so I was freaking out for my defense like months before it, and that drove me to practice it. And I tried to practice, you know, in the room that I gave the defense even. So I was just totally, you know, no surprises with the defense. And luckily it all went off without a hitch. And so yeah, I think just try to do a little bit every day, try to start early, and it'll all work itself out. I think that's also true for the science too, right? Just like you don't have to work 24 hours a day. Just, you know, do a little bit every day, keep things moving, keep the balls rolling, and, you know, keep an idea of your progress, and eventually you'll get there.
I like that. Prepare early. The best advice nobody follows, but I think it's good to still say it out loud.
I need to follow my own advice too sometimes, though, you know.
So it has been a lovely conversation with you. Thank you so much for sharing your experience with us. Before we finish the interview today, we would like to ask you one of these kind of fun questions we like to ask our guests. So probably one of the favorite questions is, if you were not a scientist, what would you be? So do you have an answer to that if you were not at the path that you are on at the moment?
Yeah, so I was on a slightly different path before I committed to science. I was actually trying to be I think like you, Jason, a physician scientist. And this is one of the reasons why I stayed in that lab with Taylor Doherty for 5 years, not only because I really enjoyed the lab and working with him, but because I could not get into medical school. I applied several years in a row, probably 100 applications, not one interview, and just rejection after rejection. And so if I wasn't doing science, if I didn't decide to do the PhD, I probably I'd probably still be applying to medical school. Maybe I eventually would have gotten in. But maybe that's a cheap answer because it's physician scientist pretty similar. But I've heard some other answers that people have said on your show before too, like being a chef. I think that was my first, if you talk to my mom and ask her what I said when I was a little kid, what I wanted to be when I grew up, it was probably a chef, which is, I think, kind of just science also, but you can eat the thing that you make at the end.
I'll also say that as a physician scientist, I don't think you chose poorly. My cup is much more full from the science side than the medicine side. There's a lot of reward in treating patients, but the current healthcare system makes that much less tangible on a day-to-day basis.
Yeah, and I am in no way bitter that I did not get in. I am very, very grateful actually, because I'm super happy with science and academia, and I couldn't have asked for a better career. And every day being so different and exciting, it's really, I think, the best job to be a scientist.
I just want to say that when I hear about how medical school works in the US, I do find it a bit puzzling sometimes that it is so convoluted and you have to go through so many hoops to apply for medical school. I hope you guys realize that although medical school across the world is usually the toughest one to get into, this is something very strange that it is something so complex and so it requires so much years of preparation and then to just not get in. I find it very, very puzzling sometimes.
And then other countries don't have loans necessarily for it either afterwards once you're in.
I mean, being a doctor in Germany, becoming a doctor is free. Most countries you don't get into, 6 figures debt to study medicine because then they want doctors that can actually practice regular medicine without having to pay their life savings back or something. I find that very bizarre, the whole system. But well, it is how it is. But science is glad that you still you decided for a PhD, and we're very happy to have had you today in the Immunology Podcast. So thank you again to Kellen Cavagnero for joining us at the Immunology Podcast today.
Thank you so much. And again, thank you for having me. And also just thank you for doing what you do. I think, the podcasting thing is so great. And I think you're doing a good service to the community for being the host of this. So good job.
Thank you very much.
That brings us to the end of our show. Don't forget to subscribe to our newsletter at www.immunologypodcast.com to get show notes, including an episode summary and links to all of the interview and Roundup Papers. You can also reach out to us on X at Immunopodcast or via email at info@immunologypodcast.com with feedback or to suggest guests. See you next time.